Hello, and welcome to a video about Immune Explorer. Immune Explorer is our gateway to SEEQ, which is the Crow Lab's next-gen sequencing database, and a set of bioinformatics tools for BCR and TCR sequences. In this video, we're going to take a look at sample selection using disease state or donor ID. Now, it is possible to manually select a run ID and sample ID based on information that you have in your notes or that you've looked up in utilities, for example. But we're just going to have a look at a feature that makes sample selection a little bit easier if you don't already have that information in hand. Within the sample selection portion of the main window, and this is similar if not identical in all the windows that interact with SEEQ, you find first the job name, and this is pre-populated just for ease of use. It points you in the right direction. This is dengue 1, which if we go ahead and enter our password and press get overview, we'll see it's going off and it's fetching data based on this run ID that we've specified here, which is 292, and one of our IGH samples for that run. All right, we've completed our dashboard job, and now you can see that the plots have been populated with some of the summary statistics. Let's get back to choosing a sample without already having the run and sample ID. The first way you can do this is using the target field, and this is populated from information in SEEQ, not from utilities. But using this, I can select Ebola, and I can see now that I've got one next-gen sequencing run for Ebola selected here. And these are the sample IDs that go along with that for my selected locus. And I can toggle through these and see that we've got a little bit of light chain sequencing as well, and no TCR data. So I can just toggle through using these radio buttons, and that will automatically populate based on samples that match my selected run ID. And for example, I know that we have quite a lot of HIP sequencing, and you can see this is populated with some run IDs. Unfortunately, not all of them. Some of the healthy data is marked as unknown, or undefined, not specified. But if we were to select dengue, we can see that we've got a more comprehensive view here. And again, almost as much light chain sequencing as there is heavy chain sequencing, but no TCR data. And so I can change this job name. All that does is when I go to run my job, I will get back a list of CSV files as well as raster and vector images that are all prefixed with that job name. And so I can clear this up. The other thing I can do is to sort by donor. And these two options can be used together. So, for example, if I were to look at donor 269, I happen to know that they've participated in a number of sequencing projects. And so I can get all the, let's see, that might not be, we're still toggled onto T-cell data, which is fine. If I switch, all right, let's clear that up and try that again. If I've got my flu time series data here, and I've got donor 269, then I can see I've filtered down the target disease or other specifier, and narrowed that down by donor, and now I've got these run IDs that are attached to it, and I can select one or more samples from that. And the only thing I would add to this is, of course, the more data you add, the longer it's going to take to produce those summary statistics. But otherwise, this is a great way to get started exploring SEEQ. Hope you have fun. Thanks a lot.